RFLP or restriction fragment length polymorphism. Restriction fragment length polymorphism is invented in 1984 by Alex Jeffreys, a technique that exploits variations in homologous DNA sequences. In RFLP analysis, the DNA sample is broken into fragments and digested by specific restriction endonucleases, and the resulting restriction fragments are separated according to gel electrophoresis. RFLP is an important tool in genome mapping, localization of gene for genetic disorders, determination of risk of disease and paternity testing. How RFLP works? Genetic polymorphism is defined as the inherited genetic differences among individuals in over 1% of normal population. The RFLP technique exploits these differences in DNA sequence to recognize and study both intraspecies and interspecies variation. First, DNA is extracted from blood, saliva or other samples and purified DNA is digested using restriction endonucleases. The recognition sites of these enzymes are generally 4 to 6 base pairs in length. The shorter the sequence recognized, the greater the number of fragments generated from digestion. For example, if there is a short sequence of GAGC that occurs repeatedly in a sample of DNA, the restriction endonuclease that recognize the GAGC sequence cut the DNA at every repetition of the GAGC pattern. If one sample repeats the GAGC sequence 4 times whilst another sample repeats it 2 times, the length of the fragments generated by the enzyme for the 2 samples will be different. The restriction fragments produced during DNA fragmentation are analyzed using gel electrophoresis. The gel is treated with luminescent dyes in order to make the DNA bands visible. Applications of RFLP. Some of the key applications of RFLP are listed below. To determine the status of genetic diseases such as cystic fibrosis in an individual, to determine or confirm the source of DNA samples such as in paternity tests or criminal investigations, in genetic mapping to determine recombination rates that show the genetic distance between the loci, to identify a carrier of a disease causing mutations in a family. Second one, SCLP or symbol sequence length polymorphism. Symbol sequence length polymorphism are used as genetic markers with polymerase chain reactions used to produce multiple copies of DNA. It is a type of polymorphism, a difference in DNA sequence amongst individuals. SCLPs are repeated sequences over varying base lengths in intergenic regions of DNA. Variance in the length of SCLPs can be used to understand genetic variation between 2 individuals in a certain species. SCLP markers are widely dispersed, selectively neutral, and their alleles typically differ by discrete steps. Third one, amplified fragment length polymorphism or AFLP. The AFLP technique is based on the selective PCR amplification of restriction fragments from a total digest of genomic DNA to generate and compare unique fingerprints for genomes of interest. After final amplification, selectively amplified fragments are separated by gel electrophoresis and visualized autoradiographically. Missile and missile fragments are excluded from the autorad because only eco-R1 directed primers are normally labeled. Typically, the autorad has 100 to 300 fingerprints with sizes ranging from 80 to 500 nucleotides. Only a subset 10 to 40 of these total bands is polymorphic between 2 related individuals. For example, Arabidopsis thailandana columbia and Landsberg erecta ecotypes. Using a 3 base pair selective primer extensions gives 128 possible linker combinations. Therefore, 128 subsets of genomic DNA can be readily amplified. Thus, thousands of markers can be generated quite rapidly.